All right, so up to this point, we've discussed from the previous lecture, we discussed glycolysis, and that was primarily occurring in the cytoplasm. A lot of the enzymes that are used during glycolysis are clustered there. We're going to be now pivoting to the aerobic respiration, and that's going to be a lot of that's going to be occurring primarily within the mitochondria, and there's going to be a few different compartments of the mitochondria we'll be situated in. And so this first slide is to try and help give everyone their help us set our bearings for where we're going to be within the mitochondria. There's going to be a few key spots that we're going to be focusing on today. The mitochondria is a double membrane organelle, where there's going to be an inner and an outer membrane, and there's also going to be this intermembrane space. In addition to that, the internal compartment of the mitochondria is called the mitochondrial matrix, and so the first after glycolysis, where we have our two pyruvate molecules produced, as we just reviewed during the quiz today, the pyruvate molecules are going to be shuttled in via these transporters from the cytoplasm into the mitochondrial matrix, where the first set of processes will occur. And then the next final steps during aerobic respiration will be involving the inner membrane, as well as this intermembrane space, where there's going to be a lot of proton, there's going to be proton pumps that are going to be shuttling these ions across. Nearly all of the steps that we're going to be focusing on today are going to be within the matrix, or right at the intermembrane space. One key thing to note is that aerobic respiration, like we said, is happening in the mitochondria, and mitochondria are only present within eukaryotic mammalian cells. Prokaryotes typically don't, and so they're going to be relying a lot on glycolysis and fermentation. And so there's going to be two major steps that we're going to be focusing on for aerobic respiration today. The first one goes by a few different names. The tricarboxylic acid cycle, the TCA cycle, also known as the Krebs cycle, named after its founder, or the citric acid cycle. It's called, the name for tricarboxylic acid refers to a few carboxylic acids that are going to be released during this cycle, and I'll be pointing out to them as that occurs. One of the key roles for the TCA cycle is the production of our reducing equivalent NADH, as well as there's going to be different points at which amino acids can be used to be plugged into metabolism to generate ATP. And so there's going to be, one thing I should say is that there's going to be, we're going to be talking about generally aerobic respiration. There's going to be a few points at which different molecules can be used to enter into these metabolic pathways, whether it's lipids or amino acids. The other final part that we're going to be talking about during aerobic respiration is going to be oxidative phosphorylation. It's also called the electron transport chain. That refers to some of the key complexes that will be transporting electrons that these reducing equivalents are carrying for us. What's one of the key critical roles is that there's going to be a proton gradient that's going to be created during the oxidation of these reducing equivalents, and that proton gradient within that intermembrane space where all these ions are going to be present, that's going to be used to drive synthesis of the ATP, and they're going to be producing quite a lot. And so so far for glycolysis, we've talked about generation of 2 net ATP. A lot of the ATP that we're going to be talking about across all of metabolism are going to be generated during oxidative phosphorylation. So as with glycolysis, I'm going to be asking you all to memorize every single molecule and enzyme. No, I'm just fine. Again, as with glycolysis, what I am going to be doing, I'm going to be asking you to take note of the inputs and the outputs, as well as a couple key features that I'm going to highlight. And so the key features that I'm highlighting are definitely things I'm going to want you to take note of. And so when I'm trying to summarize some of these slides or some of these illustrations, those summaries will try and highlight the things that I want you to take note of. So from glycolysis, we have our pyruvate molecules that we produced. And so what's going to end up happening is that there's going to be pyruvate dehydrogenase that's going to yield an oxidation event, whereby we have a reducing equivalent that's produced, as well as a carbon dioxide that's released as well. And that's going to produce acetyl-CoA. And so acetyl-CoA is going to be the input that is going to be the molecule that helps us enter into the TCA cycle here. There's going to be the key through a lot of these steps. There's going to be quite a few NADH reducing equivalents that are going to be produced. But I'm also going to take note and try and point out some of the carbon dioxides that are released, because there's going to be 3 of them. And so we have our first carbon dioxide molecule that's released up here. And so what ends up happening is that acetyl-CoA is this 2-carbon ring that's going to be used and combined with oxaloacetate here. And what's going to end up happening is that the coenzyme A part of acetyl-CoA, so there's the coenzyme A portion of acetyl-CoA, and the 2-carbon acetyl group, the 2 acetyl groups here, that's going to be combined with oxaloacetate to produce this next component of citrate. And so there's going to be a series of reactions that's going to produce NADHs, where you go from citrate to isocitrate down to ketoglutarate here. So this next reaction is going to produce a carbon dioxide and an NADH. And you can take note that every time we have a carbon dioxide molecule released, there's going to be another NADH equivalent that's going to be produced here. But it's not dependent on carbon dioxide for that to happen. And so what's going to end up happening is that there's another reducing equivalent that's not NADH, that's called FADH2. So that's another coenzyme that serves as a reducing equivalent. Bless you. And so there's only one of them here, but the key things I want you to take note of is that during TCA, that there's going to be NADH molecules, reducing equivalents, that are going to be produced. Those are key for us to use. What's key is that it's going to be used during oxidative phosphorylation for further ATP generation. That's going to be true for both NADH and FADH2. And that the TCA cycle largely derives its name from these 3 carbon dioxides that are produced. The input, I said, was acetyl-CoA, and so and I described how the two acyl groups here are going to be used with I'll take a step back, combined with oxaloacetate, and that's the input to generate citrate. And what's released here is the coenzyme A with its thiol group there. So to take note here, coenzyme A is a molecule that largely is a shuttle for acetyl groups. And those acetyl groups are going to be, for lack of a better phrase, donated to be used during the TCA cycle to enter into the TCA cycle and begin. The structure of coenzyme A is probably going to look somewhat similar to some things we've already talked about here. There's going to be a ribose sugar. There's going to be an adenine base, this phosphate bridge here. And then the main difference is going to be this pantothenic acid. That is vitamin B5. So there's going to be quite a few things that we're going to point out during metabolism that are going to end up being vitamins. And so all of these different components of our metabolic pathways are dependent on the things that we consume and eat. So take your multivitamins, for lack of a better phrase. But so the main difference here is this pantothenic acid. And so what ends up happening is it's a carrier for acyl groups here. And so when there's an enzymatic reaction that promotes the addition of acetyl groups, they'll be available here via these thiester bonds to form acetyl-CoA. What's going to be important is that coenzyme A is going to be really critical, again, for aerobic respiration here, as we've already talked about. It's also going to be really important for fat oxidation, which is how we take fats or these fatty acids and are used to enter into some of these metabolic pathways. And it can also be used in the reverse, where instead of breaking down fats via oxidation to create metabolites that can enter into these pathways, it can also take some of these metabolites and reverse and develop fat. So entry into we briefly already touched on this, but the first thing to focus on is the entry into the TCA cycle. So we have our pyruvate that we developed that we derive from glycolysis. It's going to be oxidized via pyruvate dehydrogenase, and it's going to yield the first of our 3 carbon dioxides here, and it's going to yield our first NADH associated with the TCA cycle, and that's going to finally produce acetyl-CoA that provides the acetyl groups that will be donated to enter into the TCA cycle. I think it'll come back. It should. There we go. And so the 2 carbons in the acetyl group are going to be donated to help interact with oxaloacetate to produce the next molecule in the TCA cycle, which is citrate. So the subsequent steps are not the subsequent steps that are going to be occurring to the TCA cycle. I'm not going to ask you to memorize each enzyme or each intermediate metabolite. I'll let you save that for biochemistry when you guys take that class. But what's key to note here is that there's going to be a series of oxidation steps that produce an NADH. So each step where an NADH is occurring is going to be an oxidation event where NADH is a reducing equivalent that accepts that released electron and also accepts the hydrogen ion as well. There's going to be one step going from succinyl-CoA to succinate, whereby there's going to be a GTP that's actually produced here. And what's important to know is there are these dinucleotide intermediates whereby GTP can actually be used to produce an ATP. And so there's going to be some steps in these metabolic pathways where things can kind of move back and forth a little bit. And so the phosphate group on this GTP can move back and forth across a GTP and ATP. But the key thing to note here is that definitely there will be NADHs that are produced, as well as one of these energy substrates, either a GTP or an ATP. Yeah, and the GTP may or may not, keep scooping myself, it may or may not be used to generate an ATP. So what ends up happening is that there's, I've already alluded to it briefly, but there's going to be another reducing equivalent species that's produced here. It's going to be Flavin adenine dinucleotide, and so that, or FAD. And so FAD is going to be another electron acceptor that yields FADH as another reducing equivalent here at this boxed in reaction. But there's only going to be one produced here per pyruvate. And so with each glucose, we get 2 pyruvates. So that means from each glucose, we're going to produce 2 FADH2s. And so with the final step, what's going to end up happening here is that there's going to be the last final reducing equivalent that's developed. That's then that reaction is going to produce oxaloacetate again. And so this is again, for lack of a phrase, it's a cycle that then continues to produce oxaloacetate as long as the substrates or as the inputs are available. As long as there's the acetyl-CoA to donate the 2 carbon, the acetyl here, it'll help take oxaloacetate to enter into the cycle and to produce more reducing equivalents. So FADH, again, it stands for Flavin adenine dinucleotide, or FAD. It's going to have a pretty similar structure to NADH. Main difference is that the FAD has a riboflavin, whereas NADH has nicotinamide. And riboflavin here is vitamin B2. So there's, again, another vitamin that's going to be critical to our metabolic pathways. But it shares, again, the same ribose sugar, adenine base, and phosphate bridge here. And so this depicts the difference at the riboflavin between the oxidized versus reduced form. So the complete TCA cycle here, in summary, is that we've donated, or with the input of 1 acetyl-CoA, excuse me, with the input of 1 acetyl-CoA, we were able to yield 3 NADHs, 1 FADH2, and 1 ATP. But again, as I've tried to highlight, this is all happening twice because 1 glucose yields 2 pyruvate, which means we get from 1 glucose, we get 2 acetyl-CoAs. So our net yield for each glucose molecule is going to be 4 ATP because we get 2 in the TCA cycle and 2 from glycolysis. We're going to get 10 NADHs because we already got 2 NADH, 2 reducing equivalents from glycolysis. There was going to be 2 during the entry portion into the TCA cycle, and we have 3 written here. So times 2 is going to be 6 during the actual TCA cycle itself. And we'll be getting 2 FADH reducing equivalents as well. And so this is, from the TCA cycle, this is a complete inventory of what we yield from 1 glucose molecule. And so when we enter into the oxidative phosphorylation, the next step of metabolism, the key things that are going to be used here are the reducing equivalents, the NADH and the FADH2. The electrons that they've been carrying up to this point are finally going to find their destination in the electron transport chain. Before we jump into the electron transport chain, I want to give space and leave room for a couple questions here. Yeah, Darius?
So it says that the net yield is 10 NADH.
Yeah.
But then in formula it says that 1 is
Yeah, so this is specific to what happened. The boxing part, let me go back to the microphone here. The boxing portion at the top of this slide is talking about the products during the actual TCA cycle itself. What the net yield below is talking about glycolysis and TCA cycle together, as well as like the small step in between where we go from pyruvate to acetyl-CoA, which in the in-between step, there is 1 NADH produced per pyruvate to acetyl-CoA. And because we get 2 pyruvates from 1 glucose, that means we get 2 NADHs with that in-between step. So we get 2 NADHs from glycolysis, 2 NADHs in that in-between entry step. And then within the actual cycle itself, we get 6 NADHs. And so that comes out to the total number of 10. That's how we get that number. Yes.
So we're considering the pyruvate as part of TCA.
Are we considering it part of the TCA cycle? Yes. That's part of the TCA cycle because glycolysis is how we get pyruvate. But TCA, part of the TCA cycle, or at least the entry into it, is producing the acetyl-CoA. Yeah. Yes.
So first of all, everything that's boxed in is for products during the actual TCA cycle.
Yeah.
Okay, and then
That does not include the entry step. So like this is everything that's like within the circle of the TCA cycle. There was like 1 step to get into it where we go from pyruvate to acetyl-CoA, and that yields 1 NADH or 2 per glucose. Sorry, but I interrupted you. Did that answer your question, or did you have another question? Okay. I know this isn't the most compelling lecture, but it's fairly important. And for those of you who are pre-med, this is a good entry into it before you have to study it a million times over. Yes. I already took some, but I appreciate it. Is it that obvious? Okay, I appreciate that. Are there any other questions? All right. So jumping into oxidative phosphorylation. So what ends up happening here is this is where all the electrons that these NADHs are carrying, they're finally going to find their destination here at these at these protein complexes on the inner membrane of the mitochondria. So up to this point, again, the TCA cycle is happening within the matrix of the mitochondria. You can think of the matrix as like the cytoplasm of the mitochondria. So now we're moving to the inner membrane here, where there are these inner membrane complexes. We call them complex 1 to the far left, then there's complex 3 in the middle, and complex 4. To the far right is going to be this ATP synthase or ATP pump, where the actual production of ATP will be occurring that leverages some of these hydrogen ions that are going to be moving across these complexes. So what's first going to happen is that to the far left, we have complex 1 that will first interact with NADH, where NADH now, it's been the electron acceptor up to this point. Now it's going to be the donator. It's going to be oxidized and it's going to lose its electrons. And so there's going to be intermediate carriers of these electrons that go from complex 1 over to complex 2, or to complex 3 and complex 4. But when an NADH donates here its 2 electrons, that's actually going to prompt 4 hydrogen ions to move across into the matrix here. So on the very first slide, we showed that in that inner membrane space, there was a ton of ions there. This is how those ions move across into that inner membrane space. And this is how that proton gradient is created. This proton gradient is going to be really important for this ATP synthase all the way to the right here, this pump to the far right. So the complex 1 will have, once NADH is oxidized and donates its electrons to complex 1, 4 hydrogen ions will move across into the inner membrane space. And then subsequently, there's going to be further ions moved across once these various carriers, whether they're other coenzymes or cytochrome proteins or iron sulfate molecules, are carrying these electrons over. So as they are moving over and donate across the complex 3 into complex 4, complex 3 will drive 2 hydrogen ions to move across into the inner membrane space. And finally, as well, complex 4 will also drive 2 hydrogen ions to move across in the inner membrane space here. What's key to note here is that when the electrons are donated to these protein complexes, as I'm sure you all remember from physics and chemistry, electrons have a charge. And when they're donated, and that charge is going to induce a conformational change with these protein complexes. That conformational change is what allows hydrogen ions to flow across into the inner membrane space. And finally, that buildup of those hydrogen ions are now in the inner membrane space. Are going to move down across this ATP synthase here. And I'll try and make sure I repeat this a few times, but what we have here is that for every 3 hydrogen ions, this synthase will develop 1 ATP molecule. So there's going to be a ratio of 3 to 1 of hydrogen ions to ATP during oxidative phosphorylation. But I'm gonna try and revisit that to make sure we get that ratio down. So only 10 years ago now, the actual structure for these protein complexes, or at least for protein complex 1, was properly defined. And so what ends up happening here is that we have our first electron acceptor that's part of complex 1. It's called Flavin mononucleotide. And so it's a portion of complex 1 that's actually first going to receive the donated electrons from our reducing equivalents. And that conformational change is going to actually induce these hydrogen pores to open and close. So what ends up happening is that when it's before the electrons are donated, the hydrogen pores are kind of opened up in this conformation at the top, and that's what the bottom half is, or the bottom third on the right side here. They're open at the top when they're in their reduced state, and then they're going to close when they're oxidized. Or sorry, they're going to open up when they're oxidized at the top here, and then they're going to close and open on the bottom side when they're reduced, when they gain those electrons. And so it's that donating of electrons that opens and closes these pores that allows or enables hydrogen ions to move across and get into the inner membrane space. So then the reason I should have said this on the previous slide, the reason why protein complex 1 can move 4 hydrogen ions across, whereas the others can move 2, is by nature of how many pores they have that enables hydrogen flow to move across the complex. So one of the final steps that's produced here during this oxidative phosphorylation or the electron transport chain is that there's going to be the loss of the electron that helps produce the production of a water molecule at the end of the electron transport chain. And so for those of you who are riveted and following along with everything, we've talked about complex 1, complex 3, and complex 4. I'm sure you're wondering, where is complex 2? Complex 2 is a different protein complex that's able to use FADH or FADH2. The other protein complex is 1, 3, and 4, or at least I should say complex 1 is able to interact with NADH. Complex 2 interacts with FADH2. And so what's key to note, though, is that though FADH2 is a reducing equivalent, and while it's donating electrons here to complex 2, there's no protons that are pumped across protein complex 2. So there's going to be in the instances where FADH is used here, there's going to be four fewer protons shuttled across, which means there's going to be 1.3 fewer ATP molecules developed in response when FADH2 is used here. So the last piece of the puzzle here is the ATP synthase, or this ATP pump. There's going to be this protein complex here, as well as this stock that then complex together to form the entire functional ATP synthase unit. And so what ends up happening is that for every 3 hydrogen ions that are able to interact with the synthase, that'll actually prompt the whole synthase molecule to turn about 120 degrees. And that 120 degree change is actually able to drive the synthesis of 1 ATP from an ADP molecule. And so again, we have our ratio of 3 hydrogen ions to 1 ATP via this ATP synthase here. I'm going to do 2 wrap-up slides, and then I'll stop for questions. So to review for specifically oxidative phosphorylation, to wrap up here, we took note that there were, for each NADH, there were 4 hydrogen ions that went across complex 1 and 2 at complexes 3 and 4. So that means in total there are 10 hydrogen ions for each NADH. There's going to be, because complex 2 can't drive any hydrogen ions across because complex 2 can't drive ions across, there's only going to be 6 hydrogen ions for each FADH2. And we have, again, our ratio of 1 ATP for 3 hydrogen ions. So for every glucose molecule, we have our 10 NADHs and our 2 FADH molecules, which means with a ratio of 3 to 1, with a ratio of 10 hydrogen ions for each NADH, that means there's going to be 100 hydrogen ions per glucose here as it relates to NADH. And then there's going to be 12 hydrogen ions because we have, sorry, I'm going to take a step back. We have 2 FADH molecules here, and it drives 6 hydrogen ions. So that means per glucose, as it relates to the ratio with FADH, there's going to be 12 hydrogen ions that drives across there. And so because of that, because of those numbers, we're going to get about 30 ATP, that 3 to 1 ratio of 3 hydrogen ions to 1. That means from NADH, we're going to get about 30 ATPs, and from FADH, we're going to get about 4 here, because we get 12 hydrogen ions from FADH and about 100 here. And so a 3 to 1 ratio yields 30 and 4 there. So to take a step back from the entire process of glycolysis, TCA cycle, and oxidative phosphorylation, from one glucose molecule, we're able to get 2 ATPs from glycolysis. We're able to get 2 ATPs from the TCA cycle. And then we're going to get about 30 ATP via oxidative phosphorylation from the NADHs that were developed, and 4 ATP via the FADHs. And so that yields about 38 ATP molecules per glucose. Now, this assumes that the transport of these electrons is perfect. So this is a this is a theoretical max here. The reality is that there's electron leak. There's going to be there's electron leak that occurs where not all of these electrons are going to be donated. So this 38 is a max theoretical yield, but not a realistic or experimentally derived like actual number that we would see in biology. So now I'll stop I have time for several questions here. Yeah, go ahead.
So with the electron, what happens to the components that aren't used in that run of the cycle?
Yeah, great question. That's how you can get free radical formation. That's how you can get electron leak. Because of electron leak, there's a possibility of just random free radicals be formed. And so that's where antioxidants can become really helpful. Yep.
Two questions. Do you know what is the experimental or efficiency?
It varies. Yeah, I don't have the exact number in my head, but it's not always going to be exactly the same.
So the other question is, so complexes 1, 3, and 4 exclusively deal with NADH.
Close. It's complex 1 that exclusively deals with NADH, and complex 2 that exclusively deals with FADH, because both of them will have electrons that they transport over to complexes 3 and 4. But really, it's complex 1 that is specific to NADH, and complex 2 that's specific to FADH. Yeah. Yeah.
So why is FADH used at all?
Why is it? Well, we don't have control over that.
But
like evolutionary speaking, why is there?
Why is there?
Well, there's still more. So this is a good question. So if you were to look at a mitochondria, and if you were to try and analyze the expression of how many complex 1s versus complex 2 are present, you're likely going to see evolutionary pressures that lead to more complex 1s, because we get way more, and number one, we have way more NADHs, which is also probably another evolutionary pressure where NADH is a better reducing equivalent, because it actually drives protons across. And so there's probably 2 evolutionary constraints here, where there's more complex 1 and more expression of the enzymes that yield NADH, if that makes sense. Because because again, like if it's a better reducing equivalent, there's going to be pressure to lean that way. It's not going to be like, but over evolution, it's not going to be such that like we completely get rid of FADH at all. Like there's still some use for it. It still produces ATP. It's less, but it still produces it. If it produced 0 ATP, likely over time it would be selected out, probably. If it produced no ATP, but it's still useful. It still promotes the production of ATP, just less efficiently than NADH. Yeah.
Complex 3 shuttles out 4 protons.
That's a great question. So it shuttles out 2. Complex 3 shuttles out 2. There's another coenzyme, I think it was coenzyme Q, that actually has a reaction that shuttles out 2 hydrogen ions on its own. But that does not go into our like inventory of hydrogen ions as it relates to complexes 1, 3, and 4. That's like a separate reaction.
But that still contributes to like the heat and engagement, that's the cycle.
It doesn't go into that accounting. So for, I'm wrong, it does. Forgive me.
Then in the FADH one, if complex 2 doesn't have any protons, it's like you get 6 for 3, 2, 2, 2.
Yeah. That's right. Yeah. Forgive me. Thank you. I appreciate you asking that question. So just for the recording's sake, there's a reaction depicted on the slide where there's a coenzyme Q that shuttles 2 hydrogen ions across that goes into the accounting of our total hydrogen ion inventory. Yeah. Let me see, where am I at? Right here in between complexes 1 and 3, this reaction actually produces the movement of 2 hydrogen ions as well. And so that goes into the accounting here of how many ions are shuttled across into the intermembrane space.
Is that complex?
Yeah. Yep.
Does the complex 2,3,4 pathway also have that enzyme?
Yeah. All right. So there's going to be within the membrane, within the intermembrane, many of the intermediate metabolites are fairly large and polar and require transporters to help with their movement across the membrane here in the mitochondria. And so whether it's pyruvate or a variety of our TCA cycle metabolites, these transporters are going to be pretty key and require and will sometimes require energy or can help shuttle across ATP or some of these different intermediate metabolites for their movement. I referenced really briefly that there are other molecules that we talked about throughout the course of the semester that can kind of be plugged into these cycles or these pathways. And so one example is that we can actually, in a reversible way, amino acids can be used to be plugged into some of the steps in these pathways. And so as an example, if alanine undergoes a transamination event, it actually can become pyruvate, which will then plug into our TCA cycle here. Excuse me. Aspartate, via also another transamination event, can become oxaloacetate, which is like one of the key molecules that helps enter into the TCA cycle. And finally, glutamate, via, again, a transamination event, can become alpha ketoglutarate. And so that's one of the intermediate metabolites of the TCA cycle here. And so, but this is also reversible, where pyruvate can gain an amine and become alanine. Oxaloacetate can gain an amine and become aspartate, or alpha ketoglutarate can gain an amine and become glutamate. And so this shows either the catabolism, amino acid catabolism, which is the breakdown of it, that plugs into our TCA cycle here, or the anabolism, the synthesis of our amino acids from these very same metabolites. Yeah.
Does this happen concurrently with regular metabolism, or does it only happen after progress happens?
So you're saying this has happened concurrently?
Like,
Yeah.
does it happen normally, or only
Yeah,
after
this happens normally. This is a normal part of metabolism where there are other things that can feed into these amino acids can feed into and start and initiate the TCA cycle.
So when people say your muscles break down, you know, if this the mechanism that's causing that.
This is how that happens. That they're broken. Can be broken down into their subsequent amino acids and can be used as energy substrates to create ATP and create energy. So that's the use of amino acids. The use of fats is also another substrate that can be leveraged to help create energy and feed into these pathways. There can be a series of oxidation events that allows coenzyme A to be linked onto some fatty acids here. And so as an example here, we can have coenzyme A that's bound to an acyl group that produces acyl-CoA. there'll be a dehydrogenase event that actually produces an FADH2 that produces this metabolite here. And there's going to be a subsequent, and so there's going to be a series of events where there's another final dehydrogenase event that produces this ketoacyl-CoA. So from here, we have two carbonyls. And so the beta carbon here is going to undergo oxidation that'll help lead to from 3 ketoacyl-CoA to acyl-CoA and acetyl-CoA. So this is how we can go from a fatty acid through a beta oxidation event it's called beta oxidation because it's oxidation on a beta carbon of a carbonyl to produce acyl-CoA, but also acetyl-CoA. So that means that we have acetyl-CoA that can feed into the TCA cycle here as a substrate to produce ATP through the TCA cycle and oxidative phosphorylation. And then we have acyl-CoA that can again feed back into the system. So to wrap it all up and to kind of demonstrate how some of these different pieces fit in here. And to also, sorry, another key like energy breakdown thing to focus on here is that some of the ways in which fatty acids can be stored are in the form of triglycerides here, where they have this glycerol backbone. And so there are a series of lipases or diacylglycerol lipases that can help break down some of these triglycerides. And so these lipases that break down triglycerides, that again is a way in which fatty acids are stored, it'll produce single chain fatty acids that via the previous slide, via beta oxidation, can produce acetyl-CoA that feeds into these pathways that we've talked about. But additionally, the glycerol backbone for triglyceride can also be used, whereby when it's phosphorylated, it can become glyceraldehyde 3-phosphate that can, which is a key metabolite in glycolysis. And so even the backbone for these triglyceride molecules can be used, can be phosphorylated and be used to enter either gluconeogenesis or glycolysis itself. And so what's key to note here is that if we combine all together glycolysis and the TCA cycle, there are multiple points either at the beginning where we can produce glyceraldehyde 3-phosphate that triglycerides can feed into it. It can feed into the formation of acetyl-CoA by the beta oxidation of fatty acids, or amino acids can feed into the TCA cycle with some of the different possible metabolites that can be derived from amino acids. This is a good place for me to pause and ask, are there any questions here? Yeah. Yeah, so if we try and combine together what the take-home message of the previous 2-3 slides were, the whole take-home is that, you know, we've talked about specifically glycolysis. We've been, all of you, you know, last two lectures have been having to endure looking at just pathways and hearing me talk about metabolites and enzymes and what's produced. But now we're taking a step back, and the last 3 slides have tried to show that there are other molecules that we talk about in this class, amino acids and lipids, or fatty acids, that can actually be used that can be modified via a series of reactions that then plug into this system, whereby now amino acids can help drive the TCA cycle to produce ATP, or fatty acids or glycerol can feed into glycolysis, which then can drive down and promote generation of more ATP. And so you can envision, and we're actually going to talk about this, if you're in conditions where there's less glucose, but a wealth of amino acids, that can be used to generate energy. If we're in a situation where there's a lack of glucose and an excess of lipids, of fats, that can be used to derive energy. If you're in a scenario where there's a lack of amino acids, lack of lipids, but an excess of glucose, like if you just house down a pack of Sour Patch Kids, that can be used as a source of glucose to derive energy. And so these are just trying to, this figure that we have in front of us now is going to be used to try and again, I'd like to try and revise these lectures a little bit, but what we're going to do is we're going to look at a series of different diets that are either like low protein or high fat or high carb, low protein, and like how what we've learned can explain how those diets work. Does that make sense? Yeah. Yeah. Sorry, Darius, my bad.
Can you explain the difference between beta oxidation fats and this?
Yeah, so I'll actually point to it here. So there are so triglycerides are how fatty acids are kind of like stored. And that's, they're in your bloodstream. But to go from triglycerides to fatty acids, there are lipases. Those are enzymes that just like break down fats down into like single fatty acids. To go from a fatty acid down to acetyl-CoA, beta oxidation is what helps make that happen. So to go from triglycerides down to fatty acids, there are lipases that break it down, as well as that break it down to the glycerol backbone there. But it's beta oxidation that enables going from fatty acid down to acetyl-CoA there. Are there any other questions? Yeah.
Next week, when we think on final, are we also gonna?
Yes. So yeah, since I have the recording open here, there will be a quiz it'll be even though it's like a final exam on final exam day, it'll be like a normal exam day that we've had in this class. There will be a quiz first, and if you are happy with your quizzes, with your quiz scores in this class, you are excused, and I will wish you a happy summer. If you're sticking around, you'll take the exam, which will be a normal exam length, about 30 questions, and you'll have an hour to take it. I guess we could hang out or we can do the full 3 hours if you'd like, but I don't think people will likely need the 3 hours. But does that help get a picture of what it'll be like? Good.
Clarify, sorry, I think you were meeting, right? Yeah, but it.
Yeah, the 8th. Yeah. It's not this Monday. That would be a surprise for me as well if that was the case. Very good. Go ahead.
What are the efficiency for those metabolites?
What are the efficiency rates? I don't know, but I'll look that up. All right. That it depends on what your energetics are, what you're doing. So with that you're dismissed. Let me stop recording. It's not the last thing.